how a hormone is, how it knows when to be secreted, right? When do the endocrine glands secrete these hormones and when do they know how to stop, right? So this is why I'm switching things out a little bit. Instead of going to the hypothalamus and pituitary gland, which is a little more complicated, I want to go over a very simple example of control of release, right? And, tie it to this homeostatic pathways right here. Right, so hormone secretion can be regulated, right? The endocrine glands can be regulated by signals from the nervous system, as we'll see. Other hormones can change them, or actual chemical changes in the blood, right? Can actually affect an organ, cause it to secrete more enzymes and cause its effect, right? So this would be a very simple kind of pathway, right? And so in this pathway, right, there's high blood glucose, and that all the blood runs through the pancreas right here, and the gland, right, the endocrine cells in here, they detect that stimuli, and they're going to decide to do something about it right there, right? So when we talk about that homeostatic mechanism, the stimulus is hyperglycemia, high blood glucose. The receptors are on the endocrine glands themselves. The integration center is the control center themselves, and they do the response, right? And this will go on to affect target cells, in a widespread way. And the return to normal glucose levels will stop the insulin secretion. So I'm just going to go over a little bit of this glucose pathway and see what happens here, right? So your pancreas, right? Again, it's sitting behind your liver and your stomach back there, way behind there, in your abdominal cavity back there, right? Right under your diaphragm, right under your stomach, actually. So pancreas is sitting right here. It's a complex organ. Remember I said it's got exocrine functions that secrete digestive enzymes into this area, but then it's got these little groups of cells within it that has these endocrine functions. Right? And we're going to be looking first at these so-called beta cells, right? Individual cells within here that are going to secrete insulin into the blood in response to glucose, right? So here's a group of endocrine cells. Here's all the capillaries, right? So the filtrates flowing out all around here. These cells can detect the glucose levels because of that and respond to them. All right, so here's that islet, so-called pancreatic islet with the beta cells in them. And they detect the glucose that's floating around here, says it's too high. That causes a response because it's got glucose receptors on it that take it in, right? And the response to that is to take these insulin packets that are probably already made, but maybe they make more. Maybe they're just ready to be secreted out, right, onto the cell membrane and undergo that exocytosis reaction, right? So just a note, remember, exocrine glands can do this too, but exocytosis just means a release in this fashion from vesicles. It doesn't really, it's not confined to exocrine glands. This endocrine gland does the same thing. It's just the vesicle is going to fuse with the cell membrane and release the product out into the fluid, right? Interstitial fluid, which is reach these nearby blood vessels. Right? So that's the first part of it, right? So they have the receptors on the beta cells, and they're acting as the control center, and they're going to respond to it, right? Now the hormone is going to get into the blood right here. So what happens? The effect, right, is to reach its target cells all over. There's a couple different sort of GLUT transporters, as we talked about the other day. And I talked about this in the context of increasing permeability to glucose. And now we'll get into this part over here, right? So glucose levels in this case are low, no insulin, right? When glucose levels are higher, insulin secreted, insulin reaches this cell right here. It binds to this receptor. Here we're looking at that signal transduction cascade, right? That we talked about, cyclic AMP, protein kinase A, all this. This is what you wish you just had to know about that, right? But we knew all the steps in between. And we'll just think of it as magic, right? Just magic happens right here. The signal transduction occurs. It affects these vesicles that now have these glucose transporters on them. Those go on. Says it's exocytosis. It's not really, but that's what everybody calls it because it's not actually releasing these transporters out into the open. It's just embedding them within the cell membrane. So it goes into the cell membrane right here. Now you have all these extra glucose. Well, now glucose can be taken up inside the cell. All right. So that's the effect of insulin, right? In this case, actually the one effect, right? Insulin has other effects of it here, but one effect is increasing the amount of glucose transported so that more glucose can be taken up in the cell. And these particular types are liver and muscle cells, right? So when you have excess glucose in the cell, your body wants to make sure, it's not wasted. So it takes it in. And in the case of the liver, it changes it into liver and muscle cells, they change it into glycogen and stuff, right? So in that way, you're decreasing the level of glucose in the blood and your body's dealing with that excess glucose. But this is a response to insulin, right? If you've heard of diabetes, right? There's two types of basic diabetes. One of them, it might be an autoimmune or genetic disease where these beta cells die, like they degenerate, and you lose them for some reason. And then you don't get this insulin, right? So instead of, having all these glucose transporters here, you have this excess glucose in the blood. That's why they have high blood sugar without that, right? Your glucose is there, right? You just can't get into the, cell because the insulin receptor doesn't have any insulin bound to it. There's other types. Type 2 diabetes doesn't, you do have these, you do have insulin, but there's something wrong with this whole pathway over there, right? It's a different function. All right, so this is how insulin has its effect on glucose level, right? Because once it's taken into the cell, it's out of the blood. All right, so that's your insulin, and those are coming from those so-called beta cells within this pancreatic islet. And the control is controlled by the level of glucose, as well as a couple more other complicated things, which I may or may not mention next time, right? But we're just talking about it as increased blood level glucose. Those cells detect it, release insulin. And then, lowers glucose by accelerating transport of glucose into cells, which is what we just talked about right there. There's other effects here, other downstream effects, converting glucose into glycogen, et cetera, right? Removing glucose from the blood is the bottom line here. Sometimes you have low blood sugar and you want to increase your glucose levels in your blood, right? And in this case, you have another type of cell within the pancreas, another type that secretes another hormone called glucagon. Right? And these are from the alpha cells of the pancreatic islet, and its effects are opposite, right? Its control is opposite and its effect is opposite, right? It's going to go down and to raise blood glucose level by doing these various things, which we won't necessarily get into, but they all have to do with breakdown of the glycogen or actually converting all other nutrients into glucose, right? So that's going to raise the level of glucose. Now you have more glucose floating around the blood. And now your body cells can take it in and do what it does with glucose, which is to turn it into ATP. Right. So when we're thinking about these homeostatic mechanisms, right? Again, we have all these capillaries over here. They're going to, all the filtrate is going to be in here. And if there's a lot of glucose, right, it stimulates these beta cells. Right, and which makes them secrete insulin, right, into the blood, right, which is going to travel around the body and then have all those effects right there, right? Once it does its action, your glucose is going to be taken up into those cells, your blood glucose levels decrease, right? So now when the blood flows through here, these cells say, okay, I don't need to secrete any more insulin. And then the insulin secretion can stop, right? And so at normal blood glucose levels, both of them would be rather silent, right? You get this negative feedback from from blood glucose, which was the original stimulus. If it goes below, right, hypoglycemia, right, it's going to stimulate these other type of cells, right? And we'll have a similar effect here, right? Blood is circulating around here. In this case, you got low levels, lower than homeostatic within physiological range. It stimulates these little alpha cells, which do their thing. They secrete glucagon into the bloodstream, and that does its magic, right? It basically breaks down glycogen, et cetera, et cetera. And what do? You increase glucose in the blood. Your cells take it up and do what they do. All right, so that's kind of clear about how this whole pathway might work with these hormones. Right? The insulin is being secreted into the blood, finds its particular receptor. The receptor has an action. Right? And now we can relate that to this whole homeostatic feedback mechanisms. All right, here's the other thing we just talked about, right? I like different ways. I'm a picture guy. What can I say, right? Pancreas receives a stimulus. Beta cells of the pancreas stimulated to release insulin, right? It either is going to go in the liver and muscles to take up and store it, or your body is going to take more, right? And then your blood glucose levels will decline because of this, right? And your homeostasis returns, right? So the stimulus can stop. That's your beta cells, right? Insulin, and then your alpha cells, right, are going to detect low blood glucose levels, secrete glucagon, and it's going to cause the release of glycogen, and that's going to increase your blood glucose to the point where they stop secreting glucagon. So I just wanted to kind of keep that fairly simple today, just go over the basics and go over that simple pathway, because next time it gets a little bit more complicated and it's better if you can read ahead.